Era Industri 4.0 mengarahkan pada pengembangan teknologi kesehatan menuju ke personalized medicine dan nanobiotechnology yang memanfaatkan artificial intelligence serta penggunaan mahadata dalam memberikan pelayanan kepada masyarakat. Untuk mengantisipasi perkembangan ini, jurusan Farmasi Universitas Islam Indonesia memberikan kesempatan untuk menjadi bagian dari antisipasi perkembangan ini untuk melanjutkan studi ke jenjang S2 Farmasi di UI. S2 Farmasi UI memiliki keunggulan pada pengembangan farmasi berbasis data science atau mahadata. Kurikulum ini disusun untuk merespon dinamisnya perkembangan ilmu dan teknologi, terutama dalam bidang kesehatan dan farmasi secara khusus. Sehingga model pembelajaran yang dirancang dalam kurikulum akan diupayakan dapat memfasilitasi transfer ilmu dan penguasaan teknologi sesuai tuntutan revolusi industri 4.0 dan society 5.0. Pemanfaatan mahadata ini juga akan memberikan manfaat untuk pengolahan data dalam membuat kebijakan dan meningkatkan kualitas pelayanan. Selain keunggulan utama, S2 Farmasi UI juga menyiapkan mahasiswa sebagai seorang pendidik dengan menyiapkan pembelajaran pedagogik yang diperkuat dengan kemampuan dalam menyusun kurikulum perguruan tinggi.
Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. As a participant, you are encouraged to start activating your camera and using the virtual background that has been provided by the committee. The Honorable Francois Chassagne, Pharm PhD, as our first speaker. The Honorable Dr. rer.nat APT Ronny Martien as our second speaker. The Honorable Coordinator Lecturer of Magister Pharmacy Universitas Islam Indonesia, APT Suci Hanifah, PhD, the Honorable, all the committee and all participants of Lecture Series 1 2022. Good afternoon and welcome to the speaker and all participants of the International Lecture Series 1 2022 Antimicrobial Development, held by the Magister of Pharmacy, Universitas Islam Indonesia. First of all, let us pray for gratitude for the presence of Allah Subhanahu wa Ta'ala, who has given us health so that we can gather on this day. The blessing and greetings always given to our Prophet Muhammad Sallallahu Alaihi Wasallam. Firstly, I would like to introduce myself. My name is Ilya Masyarohila. I am a pharmacy student of Universitas Islam Indonesia 2019. It's very a great honor for me to be your Master of Ceremony at this time. Ladies and gentlemen, let's start this event by reciting Basmalah together. Bismillahirrahmanirrahim. Next is reciting Holy Quran that will be delivered by Faisal Halis. For Faisal Halis, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasy syaithanir rajim. بسم الله الرحمن الرحيم يا أيها الذين آمنوا اتقوا الله حق تقاته ولا تموتن إلا وأنتم مسلمون واعتصموا بحبل الله جميعا ولا تفرقوا واذكروا نعمة الله عليكم إذ كنتم أعداء فألف بين قلوبكم فأصبحتم بنعمته إخوانا وكنتم على شفا حفرة من النار فأنقذكم منها كذلك يبين الله لكم آياته لعلكم تهتدون ولتكن منكم أمة يدعون إلى الخير ويأمرون بالمعروف وينهون عن المنكر وأولئك هم المفلحون ولا تكونوا كالذين تفرقوا واختلفوا من بعد ما جاءهم البينات وأولئك لهم عذاب عظيم صدق الله العظيم اللهم ارحمنا بالقرآن والسلام عليكم ورحمة الله وبركاته.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you and may Allah bless us. The next agenda is an opening speech from the coordinator lecturer of Magister Farmasi, Universitas Islam Indonesia, that will be delivered by APT Suci Hanifah, PhD. For APT Suci, time is yours.
Thank you very much, Mbak Ilya. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirrahmanirrahim. Alhamdulillah wa syukurillah. Wassalatu wassalamu ala rasulillah. The honorable speaker, Dr. Francois and Dr. Ronny Martien. Good afternoon and good morning in France or Spain. On behalf of Master of Pharmacy program, I would like to say thank you very much for being here to deliver a lecture for our student and also all great participants. I think this is a big appreciation for us, and it will be a very good moment for us to learn about antimicrobial development from both experts. And for your information, this webinar is as a part of a lecture of anti-infection and anti-cancer development course in Master of Pharmacy program. And this course aims to give a student understanding about basic concept of drug discovery and development. And this course is in line with our vision to build an excellent research in pharmacy area. And in this moment, I would like also to announce you that our department will implement a program of a master by research. And I think this event will be very valuable, valuable to improve, to develop, to start this program. And Master by Research will give an opportunity for students to be more focused on research. And this program have more than, I think, 75% of research-based learning rather than classroom teaching. So only the student only will have 4 until 10 credit hours of teaching, and the rest is for research. For research. So will be dominant for research base rather than teaching base. And we hope this program will bring a potential student to work more excellent research, more excellent research with a valuable publication. I think that's all for us. And last but not least, we will say thank you for everyone, for speakers, for committee, and also all participants who made this event possible. And enjoy the talks and webinar. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thanks, APT Suci, for your time. Before entering the main session, we would like to take some documentation that will be led by pharmacy staff. Time is yours. Please, for the participants, active your camera because we want to take some picture together. Okay. We will start from the first slide. Okay, ready? 1, 2, 3, smile. Okay, next slide. 1, 2, 3. Next. Okay. 1, 2, 3. Okay. And the last slide, please. 1, 2, 3. Okay, thank you. Well, now is the session that we are waiting for. For all participants, please prepare your notes and question because in this session, we will get a lot of information. The main session is a presentation from 2 great speakers, which will be moderated by APT Pinus Jumarianto and PIL PhD. For APT Pinus Jumarianto, time is yours.
Okay, thank you. Thank you, Ilya, for the time. First of all, I would like to say Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, good afternoon, and bonjour for everybody here. It's a great honor for me to be moderator here in the webinar series by Farmasi UII. Here with the topic antimicrobial development. We have 2 distinguished speakers and expertise in their field. First is Francois Chassagne from Institute de Recherche et Développement, Toulouse, France. And then the second speaker will be Dr. rer.nat Ronny Martien from Faculty of Farmasi, Universitas Gadjah Mada, Yogyakarta. I think first for this webinar, we will do a panel session here. So Francois, you will deliver your lecture first, and then after that, Dr. Ronny give your lecture, and then after that Q&A session. Okay. Okay. For the first speaker is Francois Chassagne. Bonjour, Francois.
Hi, everyone. Thank you for welcoming me into this webinar. I really appreciate that, and I'm very happy to be here with you today.
Yeah. Okay. Before you start your lecture, allow me to give a brief introduction about you, yeah, to the audience. Francois Chassagne is now is a chargé de recherche at IRD Toulouse as a research team leader specialized in ethnopharmacology. He obtained his PhD in pharmacological sciences from Paul Sabatier University Toulouse, France, and he has 22 publications, 3 book chapters, including 2 as a first author and 1 as a corresponding author, 1 book edited, and then also he has a patent, and also he got many awards and fellowships. To let you know that Francois, our audience here from various background, from staff, from student, lecturer, and then from hospital, from Farmasi, and yeah, from different background. But I hope they will enjoy your lecture here. I think without further ado, I would like to give time for you to deliver your lecture in about 1 hour. Okay, time is yours. Thank you.
Okay, so let me share my screen and present you my slides. So it's here. So do you see my presentation? Yeah? Okay, great. So my name is Francois Chassagne, so you already know that. I'm a researcher at the French National Research Institute for Sustainable Development, and I'm based in Toulouse, so it's in the south part of France. And today I'm going to talk about the research methodology and strategy for antimicrobial development from natural products. So you can see on the right side of the page some example of natural products that was discovered from a natural organism. So such as penicillin from Penicillium notatum, artemisinin from Artemisia annua, streptomycin from Streptomyces griseus, and berberin from Berberus vulgaris. So I'm going to detail that in the talk. But let's first, I'm going to first speak about the outline and the content of this speech. So at the beginning, I'm going to talk about the history of antimicrobial natural product drug discovery. Then I'm going to go over the current status of antimicrobial natural products. In a third section, I'm going to speak about the approaches for the search of bioactive compounds from natural organisms. Then the fourth sections will deal with bioassays, so biological model that we can use to evaluate antimicrobial activities. The fifth section will try to give you an overview of plants. So I'm going to focus on plants and try to give you an overview of plants with antibacterial activity. Then in the fifth section, I'm going to speak quickly about a book that we recently edited, and then I'm going to conclude this talk. So let's start first with the historical background. So the discovery of the first antibiotic was made about 100 years ago, and it was discovered by Alexander Fleming in 1928. So Alexander Fleming was a microbiologist and a physician in London. And how did he discover this this compound? Basically, he went on vacation with his family, and when he came back to his lab, he saw in one of his plates a contamination. And so, since he was a microbiologist, so he cultivated Staphylococcus aureus. So it was his main topic. He was working on Staphylococcus aureus. And when he came back from vacation, one of his plates was full of this type of colony. And at this time, he did not know that it was a penicillium colony. He just noticed that it was a blue-green mold. But he also noticed that between the molds and the aureus, the Staphylococcus aureus colony, there was a zone of inhibition. And so Staphylococcus aureus did not grow at the interface of the mold colony. So it was quite interesting for him to discover that, and he tried to make more research on this. And 20 years later, another scientist finally identified the compound that was produced by this colony and inhibit the growth of the Staphylococcus aureus, and penicillin was then discovered, and it was the first big discovery and the first antibiotic discovered in the world. So, of course, since this discovery, the golden age of antibiotic started, and it so it was from 1940 to 1970. And scientists started to screen microbes as producers of antimicrobial compounds. And so basically, they first looked at fungi. So we have seen that Alexander Fleming discovered the penicillin from a fungi, which is Penicillium. And the other scientists started to have a look at other fungi, such as other Penicillium species, but also other fungi species such as Aspergillus or other species, and they found different antimicrobial compounds such as penicillin, cephalosporin, chloromethylene, and fusidic acid. So in total, right now, we can estimate that there are more than 20,000 penicillin and cephalosporin-based molecules produced. So this is really huge, and it can show you the importance of this work on fungi. But of course, not all of them have been marketed. Only a few of them have been marketed. So the other source of antimicrobial compounds from microbes is bacteria. Bacteria was also looked at by scientists, and so scientists started to dig into the product or the compounds produced by bacteria, and they found mupirocin, monobactam, polypeptides, polymyxin. So these 4 types of antibiotics were discovered from bacteria. And I think that one of the most important species or family of microbes that was investigated, it's Actinomycetes, and especially Streptomyces. Actinomycetes are basically a type of species between fungi and bacteria, because it's a bacteria with mycelia. And Streptomyces and Actinomycetes led to the discovery of many different types of antibiotics, especially lincosamide, chloramphenicol, carbapenem, lipopeptides, aminoglycosides. tetracycline, phosphomycin, macrolides, streptogramines, and glycopeptides. So all these type of antibiotics were discovered from Actinomycetes. So I'm sorry, I hear some sound. If you can close or turn off your audio. Okay, thank you. So, and from the Actinomycetes and this antibiotic, basically it is estimated that from the 50s to the 60s, 70% of antibiotics were discovered from Streptomyces species. So you can see that not only fungi and penicillium led to the discovery of antibiotic, but it is very interesting to see that it's especially Streptomyces which led to the discovery of the main class of antibiotics. So microbes was a great source of antibiotics at this time. But other antimicrobials were also discovered from natural organisms, and not only antibiotics. So we can cite here the discovery of quinine from Cinchona species. So quinine was known by Peruvian Indians to treat fevers, and it was used for treating malaria by Europeans. And basically in 1820, quinine was isolated by two French pharmacists, Pierre Pelletier and Joseph Caventou. And many derivatives from quinine were then produced, especially chloroquine and hydroxychloroquine. And now the World Health Organization made a list of essential medicine for malaria, and chloroquine as well as quinine are included inside this list. So the other important compound that was discovered from another antimicrobial compound that was discovered from a natural organism is the discovery of artemisinin from Artemisia annua. I guess that you all know these plant species. So artemisinin, basically Artemisia annua was used in traditional Chinese medicine for fevers. And UU2 in 1972 isolated the compound that was responsible for curing and treating fevers, and especially malaria. And this compound was called artemisinin. So UU2 was a Chinese scientist. And right now, after that, so different derivatives were produced from artemisinin, especially artemeter and artesunate. And now these compounds are also parts of the World Health Organization list of essential medicine for malaria. So you can see here that not only antibiotics were discovered from natural organisms, but also other antimicrobials and especially antiparasitic from natural organisms. So when I'm going to use the word antimicrobials in this talk, I'm going to refer to antibiotic, but also to antiparasitic, antiviral, and antifungal. And of course, when I'm going to use the word antibiotic, I'm going to mean antibacterial. Okay, so let's now have a look at the current status of antimicrobial natural products. So we want to know if the antimicrobial natural products are important right now in the drug, in all the drugs that are marketed in the world, but also among the natural products and the different natural products. So I'm going to show you a result of a work that we with some colleagues, we did it with some colleagues, and we try to focus. Okay, something is not working well. Okay, we had another a look at the dictionary of natural products. So I don't know if you heard about this this dictionary. Basically, this dictionary is one of the most known natural products. I would say like one of the most known database on natural products, and it contains 200,000 natural products. So NP means natural products, right? So 200,000 natural products, which is quite huge. And among these 200,000 natural products, you have 4,000 bioactive natural products. So this dictionary was made to really inform the phytochemist and pharmacologist to know more about the natural products, and it really encompasses like the different type of natural products from different natural organism, from animalia, from plants, from fungi, from bacteria. So you can find like almost everything inside this dictionary. And so with some colleagues, we had a look at this dictionary, and we found here that so we tried to first have a look at the biological activities that are present in these natural products, and we tried to classify them and to try to see which natural products were so produced, and what type of biological activities these natural products had. So here is the biological activities that the natural products have, and also the number of natural products. And what you can see here, and this is very interesting, is that the antibacterial, antifungal, and antiviral, so basically the antimicrobial activity, is a huge aspect of the natural products. Is that most of the natural products have antimicrobial activities, and especially antibacterial. And you can see here for the antibacterial activity that it is bacteria, so in red here. So you can see that bacteria represent more than 75% of the antibacterial compounds discovered from natural products. So this is something like very interesting. And of course, here you can see that for anti-cancer, so this is the second most important biological activity found in natural products. And from anti-cancer activity, it's mainly plants who contribute for this activity. But overall, antimicrobial and especially antibacterial, but also antifungal and antiviral contribute for most of the pharmacological activity of natural products. So if we have a look at the drugs marketed in the USA, and especially so the antimicrobial agents marketed in the US from 1981 to 2019, there are in total 401 agents, so 401 antimicrobial agents that are marketed currently in the US market. And among them, you can find like 125 antiviral, 162 antibacterial, 34 antifungal, and 20 antiparasitic. And the interesting thing is that for antibacterial and antiparasitic, about half of them originate or are derived from natural products. So basically, natural products contribute and still contribute to the discovery of antibacterial, antiparasitic especially, but also antifungal and antiviral to a lesser extent. So the question is, if we have so much antimicrobial agents, since here there are like 401 agents, so you're gonna ask me, but why do we still need to discover new antimicrobial agents? And this is a good question, basically. And I guess that you all heard about the antimicrobial resistance. And this is one of the most important health issue that can really impact the world in the coming years. And a report in the UK estimates that in 2050, about 10 million people will die from antimicrobial resistance. So this is really huge. Right now, we are only at 700,000 but this is still a lot, people that are dying because antimicrobial resistance. And so antimicrobial resistance will really become a major cause of death by 2050. So basically, since there are antimicrobial resistance, the antibiotics do not work anymore. And so, of course, we need to find new antimicrobials, not only antibiotics, but also antimicrobials in general. And the other reason for looking for more antimicrobial agents is that the antibiotic market, basically right now, it's not a good business for a company and private company. It's not really profitable. Sorry, can you turn off your audio, please? There's some background noise. Okay, anyway, I'm gonna continue. It's okay. So maybe you heard about this startup specialized in antibiotic drug discovery, which is called Akaogen, and it was a company, a private company in the US. Okay, so now I'm not sure that you still hear me because there are some background noise. Okay, and so this company basically filled with bankruptcy because basically, so they discovered new antibiotics, right? But the problem is that when they marketed this antibiotic was only made for a small type of patients and only for a few patients. Since the antimicrobial resistance right now is not like huge and it's not booming, it's still, you don't have much patient to treat. So when you sell your antibiotic, you don't get back much money. So that's why they feel with bankruptcy, and then they had to stop the company because it was not profitable basically, and they did not earn enough money from this discovery of new antibiotics. So this is why we as an academic researcher, but also industrial, we need to know, we need to look and to work more in this field of the discovery of new antimicrobial agents. So I'm going to show you an example of a new antimicrobial agent from natural origin to give you, to illustrate.
Okay. Okay, sorry, Francois, there's maybe technical problem. You can continue your lecture. Are you still there?
Yeah, I'm still here. Do you see me?
Okay, yeah. Okay, please. Thank you.
Okay, thank you. So yeah, so I just wanted to show you an example of a new antimicrobial agent that was marketed from natural origin. The name is rer.nat, and it is an antibiotic approved by the US FDA. It was approved by US FDA in 2019 and by the European Medicine Agency in 2020. And this antibiotic is indicated for hospital acquired pneumonia, as well as complicated urinary tract infection and complicated intra-abdominal infection.
Sorry to interrupt you, Francois.
Yes.
Your screen, there's no, your presentation is not on the screen. Is that you have
okay.
Share the screen?
Sorry.
It's all right.
Okay. Okay, let me share my screen again. Okay, I think it's here. Yes, here we are. Okay, so I guess that's some. Okay, do you see the presentation?
Yes. Okay, merci.
Perfect. So, okay. So, Ricarbrio is an antibiotic which is based on a triple combination of 3 different compounds: imipenem, silastine, and relebactam. And imipenem is not a new antibacterial. It was discovered in the 1980s from Streptomyces, again, an actinomycetes. But here, the interesting point is that it was combined with silastine, which limits the renal metabolism of imipenem, and as well with the relebactam, which is a new beta-lactamase inhibitor. So these 2 different compounds do not have antibacterial activity alone, but when they are combined with imipenem, they increase the antibacterial effect of imipenem. So this is really important this example. I think it's really important to show you that basically we don't discover much new antibiotic, but we try to repurpose and to reuse the old antibiotics and to associate them with new drugs to improve their action. And of course, so the combination of these different drugs become more and more important to reduce the resistance and the antimicrobial resistance. So now I'm going to talk about the different approaches for the search of bioactive compounds from natural organisms. So there are like different ways to search for bioactive compounds from natural organisms. And I'm going to start first with the, I will say like the most common one, the bioprospection. So the bioprospection is basically you will randomly screen the biodiversity for new natural antimicrobial natural products. The advantage of this method is to identify unexpected bioactivity, but there are like also disadvantages. First, it's really time consuming. It takes time basically to randomly screen the biodiversity, because you don't know which pharmacological activity you will find. So you have to test your compound on different pharmacological model. So it takes time to do that. It needs a large amounts of materials. So these are the disadvantages of this method. So, for example, you can, of course, look at the marine organism. You can look at the plant or tree biodiversity or the soil microbes, and randomly pick a sample and analyze it in the lab. The second approach, which is like more specialized, is the ecological approach. And so the ecological approach basically is the field observation of interactions between organisms and their environment. The hypothesis behind that is that natural organisms, metabolites, they possess ecological functions from which a potential therapeutic use for humans can be derived. And so one of the most famous examples of an ecological approach is the discovery of Alexander Fleming, since he observed the interaction between his bacteria, Staphylococcus aureus, and the fungi, so the Penicillium colony. But zoopharmacognosy, which is defined by the observation of animals and the type of plants or natural product that they use in their daily life to cure themselves, is also another approach which is based on the ecological approach. And of course, the plant defense compounds also are inside this approach. So the third approach is the ethnopharmacological approach, and this is the one that I'm specialized in. So I'm an ethnopharmacologist. I define myself as an ethnopharmacologist. So it is my specialty basically. And the ethnopharmacology basically is based on the traditional medical knowledge. So we study natural remedies used by people of different cultures and how those medicine may work. So it's a very transdisciplinary field which combine natural sciences such as botany, chemistry, pharmacology, and social sciences such as anthropology, history, linguistics. And when we look at the methods, we can either do a field survey, and so ask people what type of remedies they used for treating specific disorders. Or we can also have a look at historical text if they exist in the medical local system. So, and thanks to these methods, we can identify some plants species, some mushroom that are used in medicine. So I'm going to present you quickly a success of ethnopharmacology, and one of the most important success of ethnopharmacology, besides the discovery of artemisinin that I already talked about. So foxglove is a European plant that you can find in Europe, especially, and it was used in the 1775 to treat edema in England. And basically, it is William Wittering, a physician from England, who really had a look at this species because he heard from one of his patients that a woman in the suburb of London used this plant to treat edema and heart failure. So he decided to see this woman, and this woman shared with him the recipe of her remedy to treat edema, and among the different plants there was foxglove. And William Wittering started to produce himself different extracts, foxglove extract, and gave administered this foxglove extract to his patient, and he basically discovered that it was really efficient and that this foxglove was really good for treating edema. So basically we had to wait about 150 years to discover the compound responsible for this activity, and this compound is called digoxin. And now, of course, this compound is still on the market. So it was approved by the Food and Drug Administration from the US in 1954. In France, digoxin is still found in pharmacy, and it is called hemigoxin nativelle or digoxin nativelle, and it is used to treat heart failure and arithmetic condition. So this is a really good example of what we can find from a plant, and not only antimicrobial, but also anti-arrhythmic and compound that can treat heart disorders. So let's go back to the approaches, the different approaches to find a new antibiotic from natural products, new antimicrobial from that natural products. The fourth approach is called phylogenetic and chemosystematic approach. So it is based on similar chemistry between similar species. So we're going to select species from genera and families known to produce compounds or compound classes associated with a certain bioactivity or therapeutic potential. So basically, we need dedicated phylogenetic tools to look for hot clades. So the hot clades are the group of species that share a common ancestry, and then when they share a common ancestry, we think that they also share a common chemistry. And so so this is the example of penicillium. When Alexander Fleming found penicillin, the other scientists started to look at other penicillium species just by thinking about this chemosystematic approach is that since there are also other species that contain penicillium, maybe they can also contain other type of antibiotic. So that's what they do. And this is also the case for Artemisia species. Artemisia species, when UU2 discovered the artemisinin, other scientists started to have a look at other species from Artemisia and try to see if they also work as anti-malarials. So finally, I'm going to speak about the last approach that you can use to discover new antimicrobials from natural compounds. And it mainly relies on in silico bioactivity prediction. And so it is computer-assisted methods. So I'm going to go over it quickly, but there are like different applications. First, you can screen a chemical library and see if one of the compounds in the chemical library in this database match with one of your model, your in silico model. And you can also do what we call the molecular docking. And in this molecular docking, the aim is to see if a ligand bind to a protein and try to evaluate the strength of this binding effect. So that's it for the different approaches. Now I'm going to talk about the different bioassays for evaluating antimicrobial activities. So basically, what kind of pharmacological model we can use to evaluate the antimicrobial activities. So I show you first the general scheme for antimicrobial natural product discovery. So as I already mentioned, so this is the different approaches that I just described before. And from these different approaches, you get the natural organism to be tested. And so you can identify the natural organism that has to be tested. And starting from that, you can then make a crude extract. This crude extract will be tested on in vitro assay. So in vitro assay are assay that we perform on cell, especially on very simple model. And then you can try to isolate or identify the bioactive compound responsible for the bioactivity that you have found inside this in vitro assay. From the bioactive compounds, you're gonna make in vivo assay and then clinical studies at the end before marketing a compound if your aim is to market a compound. So we're going to have a look first at the different in vitro assay that you can use to evaluate the antimicrobial activity. So I'm not going to speak, like I'm going to focus on antibacterial tests since I'm specialized in this topic. So I'm not going to talk about antiviral, antifungal, or antiparasitic tests, but I'm going to focus on antibacterial. And there are like basically three. Let me remind you the different living states in which bacteria can live. You know that bacteria can live in an unattached cells, unattached way of state. So basically, we also call them like the planktonic cells. And sometimes like the bacteria get attached to a surface. And so they try to develop what we call a biofilm. So this is a biofilm that is formed. And when the bacteria are inside the biofilm, they communicate with each other by secreting chemical compounds, and we call that the quorum sensing. So what you can do to evaluate the antibacterial effect is to target these different living states. And basically, you can first target the planktonic cells and try to evaluate the growth inhibition of your compounds. You can also try to see if your compound inhibits the biofilm formation. You can try to have a look at the biofilm eradication. So meaning that when your biofilm is already made, you try to see if your compounds eradicate the biofilm. And then you try to inhibit the quorum sensing. So these are the main targets that we can focus on, but there are also other targets that I'm showing you here on the right side. First, we can target the efflux pump. So efflux pumps, it is like a type of channel which extrude the antibiotic from the bacteria. So basically, the antibiotic cannot act anymore since it's not inside the bacteria anymore. And so we can try to inhibit this pump. There is also another target, which is try to make the membrane more permeable. So this is the second, another mode of action. And finally, another mode of action is to try to avoid the transmission of antibiotic resistant gene from one bacteria to another. So all this type of mechanism of targets can be used to evaluate, can be used to basically to evaluate the antibacterial activity of natural products. So I'm going to speak first about the growth inhibition that we do on planktonic cells. So there are three main types of assay: the disc diffusion method, which also call it like the agar diffusion method, the bioautographic method, and the both microdilution method. So I'm going to detail you these three different methods. Why I'm doing that is that first, the growth inhibitory assay is one of the most widely employed antibacterial tests all over the world. And sometimes we made some mistake when we perform this test. So I just want to make sure that you have the good basis and you can know more about how to perform a right growth inhibitory assay. So let's have a look first by the disc diffusion method. So it's more or less like doing an antibiogram. So basically, you have a disc which is full of a natural product. So instead of having an antibiotic here, you're going to put a natural product, and then it is put on a plate which was seeded by a bacteria. And so you can find like the bacteria all over the plate. Okay, here are the disc with the natural product. And what you can see here is the zone of inhibition. So basically, if you see a zone of inhibition around the disc, it means that the natural product have an effect on the growth of bacteria. Since everywhere else, the bacteria here have grown, right? But just around the disc, there is no bacteria. It's like the Alexander Fleming experiments, basically. So you can easily see if your natural product have an antibacterial effect or not. In this case here, the natural product won't have an antibacterial effect. So the advantages of this method is that it's fast, cost-effective, and it's very easy to perform. But there are important disadvantages that you have to keep in mind. First of all, the diffusion power of natural products is very variable, and it's not the same from one natural product to another. So you cannot compare your results based on this test, basically, because depending on one natural product to another, you won't have the same diffusion power, and then you cannot compare the zone of inhibition and the diameter of here, especially since it is not suitable for non-polar natural products. Basically, here, if you have a natural product which is like a non-polar, it won't migrate inside the agar. It won't migrate fine inside the agar. So maybe you can have a zone of inhibition, but it would be as big as the compound, a polar compound. And so you won't have, you cannot compare your results, and sometimes you can have some bias, some errors, some mistakes in the interpretation of your test. So it has to be completed with another assay. So now I'm showing you the second method, which is called the bioautographic method. Basically, here in this bioautographic method, you use a TLC. So it's a thin layer chromatogram plate with an extract. So you have a migration of your extract with the different family of compounds all over your TLC. And then you're going to overlay this TLC plate with an agar, seeded with a bacteria. And you're going to see here a zone of inhibition at a specific point, since you not all the extract is has an antibacterial effect, but you can target here more precisely the type of compound which is responsible for the antibacterial activity. And here, this type of compound is located here on this zone. So this biotrographic method is very interesting since it's fast and cost effective as well. But most of the disadvantages are the same than the previous methods. It really depends on the diffusion power of the natural products. And so it's not suitable for non-polar natural products. So once again, this second method has to be completed with another assay. And so the method that should be used, the right method which has to be used when you perform a growth inhibitory assay is this method. We call it the both micro dilution method. So basically you can use 96 well plates, but you can also do it in tubes, in separated tubes at different concentration. Here, basically what I show you is that you have here on the top the E. coli, which was seeded at the same concentration all over the plate. And here you have the Staphylococcus aureus, right? Here for E. coli, you have the natural product that was put at different concentration and in quadruplicates. So here are the same basically experiments, but in different replicates. You have another same experiments, but in 4 replicates. And basically there is a decreasing concentration all over the plate. So here you start with a high concentration, and then you dilute it by a factor 2 and another factor 2 and again and again and again until having a very low concentration of your compound. And what you can see here is that at some point E. coli won't be inhibited anymore, and you will have like the turbidity inside your well, since the E. coli can grow again, and it's not limited by the inhibition from the natural product. But here you still have this inhibition. So this is how we evaluate basically the inhibition of the growth inhibition of a natural product. And for Staphylococcus aureus, you can see that the natural product is even stronger since you need a lower concentration to inhibit the growth of Staphylococcus aureus. So this method allows comparison between natural products. So this is very important, and you can use it for polar and non-polar natural products. One of the disadvantages that maybe you need more equipment, but not so much, since you need a microplate reader for easy and reproducible experiments. But as I said, you don't have to do it in a 96 well plate. So here I show you a protocol that we have published recently with my ex-PI from the US. We basically detail and give you a very precise detail on how to perform a Bose microdilution assay. And so if you want to know more about how to perform this Bose microdilution method, you can find that online, and you can just have a look at this paper. I think it's a good base for you to start to work on growth inhibitory assay. In this protocol, we also detail how to collect and to extract plant species. So it can be also of interest if you start from a plant product, or if you want to analyze plant extract. Okay. So now that I have detailed you the different type of assay, which type of antibacterial in vitro assay should be used? So what I suggest you is to combine at least 3 different methods. Of course, the first one will be the growth inhibitory assay, and especially by using the Bose microdilution method. And then you're going to use at least 2 other methods. And all these different methods will target the bacterial virulence. And we know that by targeting the growth inhibition and the growth of the bacteria, we basically make the bacteria, we can select bacterial resistance. So it's really good to add another method that targets the bacterial virulence to make sure that we won't select a resistant bacteria, or at least not easily. And especially we can work on, you can use an anti-biofilm test, a quorum sensing inhibition test, an efflux pump inhibition test, a membrane permeability induction test, and an antibiotic resistance gene transmission inhibition test. So all these different tests can be used in addition of this first test. So I'm going to show you an example to illustrate the importance of targeting bacterial virulence. And this example is based on the Berberis species. So it was published in 2000. So it's an old article. I mean, not so old, but at least it's very interesting because you will understand that we need a combination of different antimicrobials to act on the bacteria. And that's what the plants do basically. And so they already did that. They already know how to do that. And we know that berberin, which is found inside Berberis, is an antibacterial compound, right? But one of the problems inside the bacteria is that there is a pump, so an efflux pump, the rer pump, which, so especially in Staphylococcus aureus, which extrude berberin. And so berberin cannot act anymore on Staphylococcus aureus. And in Berberis, inside the plant, you have not only berberin, but you have another compound, which is called the 5'-methoxyinocarpine, which block the rer pump. So basically, the plant species has evolved to produce two different types of compounds in order to avoid the antimicrobial resistance. So this is a very interesting example of how plant species can fight antimicrobial resistance. And I'm sure that we can find other combination of compound inside different plant species and other type of microorganism as well. So now I'm going to speak about the different in vivo assay that we can use to evaluate the antimicrobial effects of natural products. So, and I'm going to focus on antibacterial tests as well again. So, of course, one of the most famous model is the mouse model. We know it, you know it well, right? You can either make or provoke general infections, or you can also target a specific organ such as lung, skin, intestine. And you're going to analyze the bacterial load. You can analyze the survival of your mice. You can analyze the blood parameters, and you can even analyze the lesion of the skin. So this is something like very important that you can use by using the mouse model. But the problem, so the advantages of this mouse model is that it's close to human physiological conditions. So this is a very strong model and very important model. But there are like various disadvantages, especially the cost. This is expensive to use mice. It takes time. The reproduction time is very long. And there are some ethical issues mouse can suffer, of course. So we try to reduce this use of mouse model, and there are like some rules that have been established like 50 years ago already, and we call that the 3R. R means replacement, refinement, and reduction. So basically, you're going to try to replace mouse model by another model. You're going to try to reduce the use of mouse model and to refine the use of mouse model. And so to replace the mouse model, other models have been discovered, have been implemented, such as the invertebrate model. So one of the advantages is that they have a short lifespan, they are easy to cultivate, and they are inexpensive. And so these are the case for Sanero abditis elegans, Drosophila melanogaster, and Galleria menonella. So C. elegans is a worm, and it allows the high throughput screening of natural products, and it's also interesting since it has very different genetic tools available. The Drosophila so this is the fruit flies, and I'm especially working on this model since I'm developing in my lab different type of pharmacological assay based on this on the Drosophila. And Drosophila, it's an interesting model since it has a conserved innate immune response, and it also has different genetic tools available. And the last one, Galleria melonella, which is a worm. It's interesting also because it survives at 37 degrees Celsius, so you can really perform the test on the same condition than in human, and you can deliver a precise amount of pathogen. So the vertebrate model are also another type of pharmacological model, and especially the zebrafish. And the zebrafish is even better since it's even closer to human, since it has a conserved adaptive and innate immune system. But one of the drawbacks of the use of vertebrate model, or especially zebrafish, is that you need more equipment. You need aquarium and this kind of stuff to make it work. But basically, all these type of models are like alternative models that we can use to evaluate the antibacterial activity. So which antibacterial in vivo assay should be used? So of course, what I suggest you is to use a preliminary test on invertebrate or vertebrate model, and you can select one model among these 4 different type of models, so either Drosophila, C. elegans, Galleria melonella, or zebrafish. And then when you have a lot of you know more about the mechanism of action, you know more about the activity, you can start to validate and to confirm your this antibacterial activity on a murine, on a mouse model of infection. So yeah, this is basically what I suggest you to do for in vivo assay. So now I'm going to focus more on plants and plants with antibacterial activity. So I'm going to give you an overview of plants with antibacterial activity. And this discussion is based on an article that we published 1 year ago with some colleagues, and we did a systematic review of plants with antibacterial activities. And so this is a huge work because we had to review 9,000 articles in total, and we based our review on 3 different databases, electronic databases, Web of Science, PubMed, cFunder, and we used plant inhibitory concentration and antibacterial as keywords. So we had like 9,000 articles we have found 9,000 articles, and we screened 4,000 different articles. So we were like a team of 15 people to review all these articles, and we had like very strict criteria to review these different articles. And at the end, we included inside the review 483 articles. So it really gives us an overview of the different characteristic features of plants that was basically used or investigated for their antibacterial activity. So the most investigative botanical groups for antibacterial activity were, so basically we're going to start first with the most investigated botanical family. And we can see here that most of the species that was investigated come from like very famous and botanical groups, Lamiaceae, Fabaceae, and Asteraceae, which are also like the 3 more important group of botanical family all over the world. The most investigated botanical genera were Acacia, Cinnamomum, and Salvia. So you can see here that these 3 different genera were highly investigated for their antibacterial activity. And finally, when we look at the plant species, it is Cinnamomum verum, which was highly investigated, followed by Rosmarinus officinalis, Thymus vulgaris and Origanum vulgare that are especially present in the Mediterranean region. So when we look at the part of plants most investigated, people had especially a look at the leaf and the aerial part. So that was the results that we got. And when we look at the types of extract that were investigated, it was mainly crude and methanol and crude ethanol extract that were investigated. Regarding the different bacterial species that was targeted, we can see here that Staphylococcus aureus were the most investigated species. So basically, the scientists, when they performed their test, they used mainly Staphylococcus aureus to prove the antibacterial activity. And in a second, they also used E. coli as another target, but Staphylococcus aureus was really highly employed. So this can be something like interesting, since you know now that Staphylococcus aureus has been widely targeted. So maybe it could be interesting to target other bacteria than just Staphylococcus aureus and E. coli species. And so if we have a look at the plant species in clinical trials for infections and inflammatory disorders, I'm going to show you like 6 different species that has undergone that are in clinical trials for infectious and inflammatory disorders, and so that represent like a very huge source of anti-infectives and antibacterial compounds. So first of all, we have cranberry. So cranberry, also called the Vaccinium macrocarpon. It is especially used for urinary tract infections, and 40 different clinical trials have been performed with this extract. Echinacea purpurea was also highly investigated. 22 clinical trials have been done, and especially by targeting respiratory tract infection. Aloe vera with wounds was also a huge target. Licorice, Glycyrrhiza glabra, with 17 clinical trials have been especially investigated for the caries prevention. And pomegranate, Punica granatum, 12 clinical trials for different disorders, especially rhinovirus infection. And finally, curcuma longa, 10 clinical trials for infections and inflammatory disorders and different type of disorders. So we already know some of the compounds from these different, some of the bioactive compounds or the antibacterial compounds that are active. For cranberry, proanthocyanidin is one of the compounds, but it does not have an antibacterial effect on E. coli, but instead it avoids the bacteria to attach to the urinary tract. So this is something also important, that you may not find an antibacterial effect, like a growth inhibition effect, but you may find another type of mechanism of action. So that's why you need to use different type of antibacterial test, at least, to evaluate the overall antibacterial activity of the compound. For licorice, you have glabridin, which is also another antibacterial compound, and finally curcumin from curcuma longa. But the most important thing, and we have seen that from berberis plant species, is that it's mainly a synergy between compounds. Inside the plant, inside one plant species, you have different compounds that act with each other, and that makes this synergy and this antimicrobial effect. So I'm going to go over the last section, and I'm just quickly give you a short introduction to a book that was recently published. So it's a book that was published 1 month ago. So that's why I thought that it could be good for you to know more about it. And it's a book on medicinal plants as anti-infectives. So in this book, 17 chapters have been published. So it's more than 600 pages. So people from different countries have contributed to this book. 13 countries, 58 authors from 13 countries. There are like 2 sections. The first section describes the antimicrobial from plants in the world, and the second section, which is I think very interesting to you, is that it describes new tools and innovations to study antimicrobial medicinal plants, especially metabolomics, DNA, barcoding, retrospective treatment outcome study. We also give some information on chrome quenching and biofilm assay. So I really encourage you to have a look at this book. So it's a very valuable resource for students in pharmacy, but also in ethnobotany and medicinal chemistry, but also for scientists and researchers, especially. And you can have get a preview on Google or book Google online of this book. If you don't want to purchase it, of course you can have a look at the book online freely, and it's also available for purchase on Amazon. Okay, so I'm gonna conclude this talk now. So the main ideas that you should keep in mind from this talk is that, first of all, we don't discover so much antimicrobials right now. The golden age of antibiotic is over, and there are less and less new antimicrobial scaffolds discovered each year. So it becomes a real issue. And one of the things which is really positive is that the biodiversity, as we have seen, can be a huge source of new antimicrobials. And it has still to be explored, and there are a lot of things to explore, especially in Indonesia. I think you have like different types of biotopes. You can focus on marine organisms, on microbe soils, on forests, on traditional knowledge. So there are like a lot of things to do, and I'm sure that you already did a lot of things. And if you need help, I'm here to help you, of course. But there are still many things to discover. So we need more researchers in academia to enroll in the search of new antimicrobials, and this is the reason why we need more training. And this is really important that you follow this different webinar, and because this is how we can basically fight the antimicrobial resistance and train more researcher to fight this antimicrobial resistance. And of course, to fight this antimicrobial resistance, we need to use and develop innovative tools, and especially new pharmacological model and new mechanism of actions. And so we need to collaborate with each other to make sure that we have all the different tools that we need to work on the antimicrobial resistance issue. Okay, so this is the end of my talk. I thank you very much for your attention, and I hope that you liked this talk. Thank you very much.
Thank you, Francois, for your really great lecture. So it gave us like enlightenment how the antibiotics and antimicrobial that and then the current position of the research of the antimicrobial, which is actually now it's kind of not profitable for the industrial pharmacy, if I'm not wrong. And then it is interesting because we need actually antibiotics, as we know that the antibiotic resistance is still the issue until now. All right. I think we like to move to the second speaker here. The second speaker will be Dr. Ronny Martien. But before that, I would like to give a brief introduction of Dr. Ronny Martien. Good afternoon, Dr. Ronny Martien. How are you?
Hello, yeah, good afternoon for you as well.
Okay. First, I would like to give you a brief introduction of Dr. Ronny Martien here. Dr. Ronny Martien obtained his doctoral rer.nat in natural sciences with an excellent pass with distinction at the Department of Pharmaceutical Technology, Institute of Pharmacy, Leopold, France, and Universitas in Group. He got many scholarship from bachelor degree until a postdoc program. He has more than 70 publications, and then he wrote a chapter book in oral delivery of nucleic acid drugs. He received many awards as well. He has 10 patents here, and also he already conducted more than 50 projects since 2011. So he's really productive. Yeah, and his expertise in nanotechnology. So it's a great honor to have you here, Dr. Ronny Martien. And I think without further ado, I would like to ask you to deliver your lecture with the topic, the approach of nanotechnology in development of the antimicrobial agent. The time is yours, it's about 1 hour. Thank you.
Yes, thank you, Mas. Thank you me to share, maybe to share my knowledge, to share my experience, things about nanoparticle, especially for fighting or combating with antimicrobial or antibiotic issue in Indonesia. And good afternoon for everyone, for the participants who waiting and still listen to this webinar. I will ask for share my okay. Is it clear? Or everyone can see my PPT?
Yes, it's clear.
Okay, maybe I don't want to take long PPT or about how to combating antimicrobial issue, and I will make I will make disclaimer. I'm not apotheker. No, I don't have any degree with APT, but thank you. Today I have it, and basically I'm microbiologist, so I'm working along with bacteria during my bachelor during my master degree. I work a lot with bacteria, and then for my PhD degree, I move a little bit to nanoparticle. But in this slide, we can discuss or maybe we can share how we can deal with antimicrobial or antimicrobial issue. So my name is Ronny Martien, and my talk is about the approach of nanotechnology development of antimicrobial agent. And I'm staff and lecturer, also researcher and pharmacy department at Farmasi Faculty of Pharmacy, Department of Pharmaceutical Technology, Gadjah University.
Apa efek calcitriol pada sel-sel imunitas? Sel-sel imunitas yang di
This is my campus. Maybe everyone in Jogja knows about UGM or Universitas Gadjah Mada. So, as a tropical country, we are live in very good condition. Every day summer, 37 or at 34 degree, always warm, and humidity is rise between 80 till 90%. And this is good condition for bacteria to grow, you know. So when we live in Indonesia, we surrounding by bacterial actually. So, and when I was a child, well, my mom told me we just lucky, you know, living in Indonesia or in tropical. Because we are getting stronger, because as a child we just let they just let them us playing around with dirt, with playing around with sand, with everything that maybe is not sterile, you know, and give us a good immune system for deal with bacteria or microorganisms around us. So, and there is, of course, there is a good and bad things for the bacterial around us. The good things for microbial or bacteria, maybe you can make for food, you can make for yogurt, you can make for fermentation product, but some of them is harmful for human, you know. Especially in this picture, yeah, maybe it's not including virus, but a lot of bacteria also dangerous or harmful for human. And so when we get infected by some bacteria, of course we need a drug to combating the bacteria in our body. There's some, let's say, pathway how antibiotic, antimicrobial drug can kill the bacteria. So, for example, they destroy cell wall. In this case, they say beta-lactam, penicillin, cephalosporin, monobactam, carbapenems, and so on. They kill the bacteria by destroy or disturb the cell wall. And also they also has a pathway to destroy or disturb plasma membrane like polymyxin B, colistin, lipopeptide, and tomycin. And some of them also have a pathway to destroy or to kill bacteria by interfering their DNA or RNA synthesis, as for example, fluoroquinolones, moxifloxacin, and rifamycin. They kill the bacteria as antibacteria with destroy the DNA or interfering DNA synthesis or RNA in their cell. They also have effect to block the protein synthesis with destroy the ribosomal cell. For example, they destroy as subunit like tetracycline. They destroy 50 as subunit. They destroy the interfering lymphocytes, and also the rest. They also can kill the bacteria by interfering metabolic pathway. For example, folic acid and mycolic acid. This is for this for this slide. You can see some action or mode of action of antibacterial or antibiotic with how they make action, the target and drug class. Well, like I mentioned before, first it inhibits cell wall biosynthesis, it inhibits biosynthesis of the protein, disturb the membrane, inhibit nucleic acid synthesis, and anti-metabolite, and microbial adenosine triphosphate synthesis inhibitor. So this kind of mode of action is happen when we give antibiotic or antimicrobial in our body, and that drug meet the bacteria and kill them from our body. And we have a big problem with antibiotic now. They become resistant, you know. A lot of germ and some are drug resistant. When we apply the antibacterial and the patient doesn't apply the dose of the antibiotic, one of the bacteria maybe still survive, and once bacteria survive and they can make double time and then make reproducible. They make double time, and then they will grow as a super microbial, we can say. And we can just when we want to fight the resistance of the bacteria, we cannot just always change the antimicrobial that we apply for the patient, because like Francois mentioned in his conclusion, but because reduction of the new, because now we have a reduction of the new antibiotic discovery, so less and less we can find the new and the new entity drug for combating antibacterial. So when we always change the active ingredient, active drug for combating antibacterial, we cannot racing with the resistance of the bacteria. So we have to hand by hand with another method to combating the antibacteria. For example, with drug delivery system that I apply now. So how in this slide we see there is some mechanism that how the antibiotic that we apply to the bacteria cannot kill or destroy the bacteria. For once, because there is the alternation of the target site of the antibiotic, so the bacteria somehow can change the active site or target site of the antibiotic. So when we apply one of the antibiotic to the bacteria, somehow they cannot recognize the active site, and the bacteria. The antibiotic can have any damage to the cell wall or to the membrane cell. The second, also they can produce enzyme that inactivate the antibiotic. Bacteria also living thing. They always learn, always see or learn by what happened with their bacteria. They can also, because they also a natural defense for the bacteria, they always try to find how survive when antibiotic applied to them, and they also can produce enzyme that can activate the antibiotic. The third, the alternation of in the cell membrane resulting the decrease of permeable just decrease uptake of antibiotic. Yeah, they also can change the permeability of the membrane. So the antibiotic that used to be can deliver to the cell of the bacteria somehow is blocked because the the permeability of the cell or wall, cell wall or cell membrane of the bacteria change, become stronger, so the antibacteria or antibiotic cannot enter the cell. The fourth, number 4 is by removal of antibiotic using active transport. That in this case we call efflux pump. So efflux pump is like a natural mechanism also for the cell, for the bacteria also to efflux or to what called to pump out everything that enter to the cell that they feel that is harmful for the cell. They will pump out out of the cell. So whenever we apply the antibiotic to the bacteria, maybe they can enter the cell, but somehow a efflux pump can pump out again to the out of the cell the drug. So the activity of the drug will be decreased, and then the bacteria will be survived. And then the last, how the bacteria can survive from the antibiotic is somehow use the alternative pathway which can compensate for the action inhibited by the antibiotic. So they change the metabolism of the cell inside the bacteria, so the antibiotic that we apply to the bacteria can have any effect. So this is like the problem or the mechanism. Maybe it's good for the bacteria because with this mechanism, bacteria can survive. But for human that can fight against that bacteria, maybe it's not good point. So this is like our problem, how we can tackle this mechanism, so we can and then we can find or we can produce or formulate the new antibiotic for formulation, maybe with the same antibiotic, not with the new one, but with another target or another drug delivery system. Yeah, this is for. I try to make a small conclusion, but with the bacteria, they have efflux pump. So when the antibiotic enter the cell, they will pump out the antibiotic out of the cell. So this always like this, because like I said before, this is a natural standard or natural defense of the cell of the bacteria. When they find strains or something active ingredient that they don't like, they will come out to the cell. The second one, they have a plasmid or in DNA level, they can that plasmid or DNA they can upregulated antibiotic resistant gene, so they can produce or they can have the genetic that can have resistance to some antibiotic. The third one, that for the small conclusion, they can produce antibiotic degrading enzyme. Sorry, so they can produce. Because they always learn. They always learn whenever they find some active ingredient, some ingredient that they cannot recognize in the cell, and then they try to figure out what is inside. So they learn, and then they can produce antibiotic degrading enzyme, and that enzyme and then can cut or maybe to block or maybe to destroy or inhibited antibiotic effect. Okay, this in general, I think we we all know there is a two big group of bacteria, is gram-positive and gram-negative. The gram-positive bacteria usually stronger against the antibiotic than gram-negative bacteria because they have a thicker or more layer of peptidoglycan than negative bacteria. So this also we have to keep in mind because we always, because these two group of the bacteria is important to measure how much or how much doses that we can give to the patient when we have to compare with two group of these bacteria, which is gram-negative or gram-positive bacteria. Of course, with gram-positive bacteria, we need more maybe doses than gram-negative bacteria. So, what's nanotechnology can take a part in this battle, you know, because Francois already told you, but they try to find a lot of ingredient, a lot of active compound came from a lot of plant, maybe from all over the world, to fighting the antibiotic issue or antibiotic resistant issue. Because I said myself, is like I'm working a lot with nanotechnology. So nanotechnology can produce nanoparticle. This in general, nanoparticle can inhibit cell wall synthesis, can make oxidative stress, and then they also can inactivate protein synthesis. They can also inhibit activity enzyme that enzyme can block the activity of antimicrobial. They also can make targeted signaling to the cell. Nanoparticle also can modify essential protein inside to the cell. They also can, nanoparticle can also help protein or drug which has antibacterial activity penetrate into the cell membrane. So I will discuss a little bit with more slide afterward. So yeah, this is antimicrobial agents. We already knew from speaker before, from Francois, that there are a lot of antibiotic as antimicrobial agent of antibiotic against from plant, from synthesis, maybe from the sea and so on. But I will try to define with 4 mechanisms how nanoparticle can help antimicrobial agent take a part fighting with antimicrobial or antibiotic issue. First, when we talk about antimicrobial issue as a metal, for example, this is like a silver argentum metal as usual, you know, because when we talk about in Indonesia we call perak, perak or argentum is famous for antibacterial activity. So before human find antibiotic as penicillin at 1928, when Perang Dunia Pertama atau World War happened, they all use a lot argentum for curing the wound, because argentum has activity for antibacterial. This without nanoparticle, the argentum cannot be stronger as antibiotic as now, because we cannot formulate the metal, for example, this para to be formed as what call it gel for curing the wounded. So this nanoparticle, for example, for this article, I can show you that the antimicrobial mechanism of silver and application in dentistry. So how the silver can combat or can fighting against bacteria in the term of antibiotic. First, the silver can disturb the cell wall and membrane by argentum silver positive ion. So with that positive ion, they can attach to the cell, to the membrane of the cell, and then disturb the cell membrane, and then they can make the membrane more permeable for drug. So when the membrane get more permeable, and then the drug or the antibacterial can enter the cell easily, and then kill the cell or kill the bacteria. The second, after after they disturb or they make the cell membrane more permeable, the silver also can block or can interfering the organella cell, such as a ribosome. They block 50S and 30S organella of the ribosome, and then the synthesis of the protein of the cell will be stopped, and then at the end they also can kill the bacteria. And then they also they can interrupt the production of the ATP as a source of energy of the cell. When the cell doesn't have ATP or doesn't have energy to support their life, they also can kill the cell or the bacteria. Number 4, they also, like number 1, they also can disturb membrane by reactive virus or reactive oxygen species. They somehow to disturb or to unstabilize the membrane of the cell, and then the bacteria can kill by there are a lot of oxygen can interrupt into the cell. Number 5, after they also can interfere DNA replication, and then DNA replication can stop, and then the cell will be die. Number 6 is by denaturating membrane. Silver nanoparticle, when the membrane is gone, and then bacteria will be lysis and then dead. The number 7 is by perforate membrane by silver nanoparticle. Beside of silver, we can also produce another metal nanoparticle. For example, with gold or gold nanoparticle, Seoul nanoparticle, argentum nanoparticle, as I mentioned before, with ferro nanoparticle and zinc nanoparticle. So with this nanotechnology, we can make small, effective single nanoparticle from the metal that can have activity that may be useful for combating antimicrobial issue or antimicrobial resistant issue with our society. So okay. The other strategy is also how we also can encapsulate the active site of the we can also encapsulate active ingredient or antibiotic that can be applied to the cell, because for example, when there is a mechanism that the bacteria can change the active site of the target of antimicrobial, when they encapsulated the active ingredient, or when we encapsulated the antibiotic, and then they can recognize it anymore. And then this encapsulated antimicrobial can easy to attach to the cell, because this usually they have a positive charge polymer, and then when they enter, when they attach to the membrane of the cell, they can induce the what called endocytosis, and then the antimicrobial drug will be swallowed by the cell and then enter to the cytosol, and then will release the drug inside the cell. And then whenever the drug getting sent to the cell, they will acting as antimicrobial activity. So this also we can see from the journal we title preparation of characterization of kanamycin nanoparticle, ketosane nanoparticle to improve the efficiency antimicrobial activity against nosocomial pathogens. So in this journal they try to avoid the recognition of bacteria with kanamycin as a model drug. So they make kanamycin as nanoparticle combined with chitosan, because chitosan also biodegradable polymer who has a positive charge. Is it important because cell membrane have negative charge? Whenever we make nanoparticle with positive charge, it's easy to the drug or to nanoparticle to attach to the cell, and then induce the endocytosis. So how to make this nanoparticles? Quite easy. We just mix chitosan with sodium triphosphate to make a cross-link, to make a nanoparticle. We just sidder it and then add a kanamycin as a drug, and then we can measure, maybe if we want, we can measure the drug loading of the kanamycin inside the nanoparticle. And then we also, and then when the kanamycin chitosan nanoparticle and then chitosan nanoparticle itself going to in vitro study, like Francois mentioned before, we had just make a plate. We just make a plate agar. We pour the bacteria that we want to test. For example, we use bacillus or E. coli, for example, and then we make some dots or like wells inside to the plate, and then we apply or we put Cantosan nanoparticle as a control and kanamycin nanoparticle as a model drug, we can put it together and then we measure the hollow zone of the cell. And as mentioned in this journal, that with nanoparticle, kanamycin nanoparticle, the effectivity of the antimicrobial activity is more pronounced than the control. I think Francis also mentioned almost of the antimicrobial drug has a problem with polarity. Almost of them have non-polar, so they cannot easy to travel from the well out to the agar, because agar or agar of the bacteria is made by water based, so they cannot easy because they all somehow will be precipitate. So with nanoparticle we can change maybe the solubility of the drug from non-polar become polar. I think it's not really change the solubility, but we make like we make the behavior of the nanoparticle in the water will be more better. So this is also one of the mechanism. We encapsulated nanoparticle with nanocapsule made by polymeric nanoparticle, or we can make with, for example, with nano emulsion, and then we can also produce or formulate to make a controlled antibiotic to combine. So nanoparticle can penetrate into biofilm. That it is also a big problem when they start to make biofilm. Somehow the antibiotic cannot enter to the core of the biofilm, so they can have effect. But with nanoparticle, they can penetrate into the biofilm, and then nanoparticle can help antibiotic to diffuse into the membrane cell of the bacteria, and then of course they will destroy the cellular component and then make the bacteria is dead. The other one is make like the same like and with antibiotic loaded polymer, like the same like polymeric nanoparticle, and then like I said before, they can attach to the membrane of the cell, and then the cell, because like I said, cell has negative charge and nanoparticle have positive charge, they will attach and then somehow can induce the endocytosis, and then when the nanoparticle getting inside the cell, and then will release the antibiotic, and then can have effect of the antibiotic. Antibiotic can increase the penetration, antibiotic target and destroy the cell, and then of course, and then reduce the cell efflux, and then at the end will be make the cell dead. The other one, the other component of antibiotic agent is protein or antimicrobial proteins. For example, in this slide, this we call manganine. There is indolicidin, protocrin, and defensive. This is a small peptide that have effect for antibacterial. So is it because antimicrobial protein has a problem with not with toxicity, but with stability, you know, because this IMP is a protein, so the EC can be degraded. Maybe it's not really cut by piece, but as we know, protein has a three-dimensional property. Whenever they change a little bit of the three-dimensional form, the effect of the antimicrobial protein will be gone. So we have to protect it, this IMP before they deliver inside to the cell. So we cannot just apply the EMP naked to our body, because our body have a body fluid that can destroy easily this antimicrobial protein. So we need to protect the EMP first before they can reach the target to the bacteria. The easiest one is with nanoparticle, because with nanoparticle, like as before, is like encapsulated. So they encapsulated the AMP as protein, so they can help the drug, in this case AMP, inside the cell, and then can release the drug inside the cell, and then can have an effect inside to the cell. So nanoparticle help the AMP survive from the outside of the cell, and then getting inside the cell to get any effect of the antimicrobial properties. So this picture with antimicrobial peptide, we can also combine with carbon nanotube. So carbon nanotube is, when we saw the picture of carbon nanotube, is just like straw that have compartment in the middle that we can load the antimicrobial peptide inside the carbon nanotube. Because we can also use solid lipid nanoparticle. We can also use micelle, liposome, SNAP, dendrimer, and hydrogel. So this kind of vehicle can be used for antimicrobial peptide, help to get more better action to kill the bacteria, because they help the AMP getting inside the cell, and then by disruption of the membrane, and then at the end they can kill cell by their activity. That is also so this picture is also talking about EMP with different little bit different system, but almost the same as a slide before. So the last one is exchange stability. I just typed this journal from the internet. The title is Enhanced Stability of Activity of Antimicrobial Peptide in conjunction with silver nanoparticle. We also can combine this AMP with the silver nanoparticle, because as mentioned before, with silver nanoparticle can somehow disturb or destroy, or disturb the membrane cell or cell walls, and then they can make the cell membrane cell more permeable, and then let the antimicrobial protein getting inside the cell, and then get effect of their properties. Yeah, this also happy with encapsulated with another nanoparticle, as mentioned before, because the issue is still the same, because the issue is how to make MP can apply to our body with good stability properties. So the last method is by oxidative stress. Usually, in this case, we use with metal nanoparticle, like silver nanoparticle that I mentioned before. We can also use Cu and then gold nanoparticle or fair metal nanoparticle of ferro, because they have stress, oxidative stress, and with that metal they can also cell the image of the cell, and then also have they can also lock protein synthesis. They can also destroy the DNA. They can also change the permeability of the membrane. They also change or lock the efflux pump, and then at the end they also can kill the bacteria. This is the publication that I take, antibacterial approaches in tissue engineering using metal ion and nanoparticle from exposure to application. In this figure, the field emission scanning, we can see from scanning microscope electron that E. coli and Staphylococcus aureus as a model bacteria. We see for the figure C the cell availability of Bacillus and E. coli after treatment with negative iron oxidano particle and positive iron oxide nanoparticle in this at different concentration. So in this figure with nanoparticle, it have a different effect, or let's say have a better effect for killing the bacteria than without nanoparticle. So in this figure, we also can conclude that with nanoparticle, the metal or ferro or iron working more pronounced when they combat or they have to kill the bacteria. So this is my talk. So I just want to give some acknowledgement, and then this is my group research, quite big group research, because we have a lot of grant, a lot of application, of course. And okay, let's start working with antimicrobial nanoparticle. Thank you.
Okay, thank you, Ronny, for your great lectures today. It gave us more insight to how the nanotechnology can help maybe the problem that has for the antibiotic formula here, especially maybe there is some formula that can help with the nanoparticle size. All right. Now we are going to have a Q&A session. We have maybe about 45 minutes until 4 o'clock. So we will have like maybe 2 sessions. I saw there's one participant that has raised her hands. Nur Aini Ihtikafsari. Do you have any questions to the speakers? Nur Aini, are you still there? It seems maybe it's kind of accidentally raise a hand maybe. Is there any questions that's from audience directly to the all right, there is a hold on. There's a question in the chat box actually from Ibu Asih Triastuti. She want to ask to Francois. As we learn from the pandemic COVID-19, there are several predictions said that we may face another pandemic in the future. In your opinion, what are the approach for antiviral agents development? Is it similar for antibacterial agents? To Francois,
Yeah, so that's a good question, really good question. I guess we are all afraid of this pandemic, and we are scared that it will become bigger and bigger. Yes, so it's quite different, antibacterial tests and antiviral tests. Basically, since bacteria can live, you know that bacteria can live alone, of course, with nutrients, but it does not have to live inside the cell. But virus, it has to live inside a cell to replicate itself. So you won't use the same test, of course, and you will focus mainly on the cellular machinery, so the human cellular machinery to target, to develop different targets to avoid the virus to replicate inside the human cells. So there are like different possibilities and different targets, of course, inside the human cells to avoid that the virus replicate itself inside. So it's quite different, and you need molecular biologists. You need molecular biology to help find the solution for this antiviral agent. And there are also something which is interesting, and we have seen that through the pandemic, is that not only antiviral agents are necessary to combat this type of pandemic, but we also need how can we say, like confinement, or the fact that we don't see each other, and we also need vaccination. We also need, so there are like different way to avoid the pandemic, and not only by focusing on antiviral agent, since it takes time basically to develop a new antiviral agent, even if it's a new antibacterial agent, you know that it takes time. So in this case, when there is something that happen quickly, and we need antimicrobial agent quickly, sometimes it's better to look for other options in the time we have, and before reaching the antiviral and developing antiviral agents.
all right. Thank you, Francois. Bu Asih, I would like to confirm. Do you have any more questions to Francois to maybe to confirm, or maybe more questions to Francois?
Okay. No, thank you, Pak Pinus. Thank you, Francois.
Thank you, Francois. Thank you, Bu Asih. All right. There's one question from chat box here from Uzul Hikmah here. Allow me to in bahasa Indonesia, Francois. so I think I should translate it to you, but maybe for Pak Ronny first ya. Sediaan nanopartikel dapat meningkatkan kelarutan dan bioavailabilitas obat, tapi katanya pernah berbanding lurus dengan peningkatan efek samping obat. Jadi bagaimana untuk mengatasi permasalahan ini? Mungkin saya harus translate sedikit buat Francois. I would like to translate the questions. The nanoparticle dosage form preparation can increase the solubility and bioavailability of the drug. But in the literature, also it's said that it is equal to the increasing of the side effect of the drug. So how to solve that problem in the development of the nano particle dosage form?
Okay, should I?
Yeah, please.
Okay. Yes, of course. You know, when I just have the analogy when we use knife. You know, when you knife and then we sharpen the knife and then we get sharpened knife. You know, it's good, of course, but also dangerous. You know, it's the same. You know, when everything in the world is always bad thing and good thing always side by side. You know, when you good, the bad things also that big. When we talk about drug, of course, when we drugs or when drug is basically is toxic, because when we saw the logo of the Farmasi is snake poison, you know, because it's toxic. But we have term of doses or dose. So we, of course, whenever we want to apply the nanoparticle to the market, for example, right, to have a product, for example, we have make sure that the product the product itself doesn't have any effect or have any toxicity, for example. So of course, and then we make a toxicity test with cell, for example, and then next level we have toxicity with rat, with acute toxicity, and then we apply if the toxicity with several doses is safe, of course, and then we can apply for clinical trial phase 1, for example. Yeah, of course, the drug itself, when for example, water, water when something when we drink now is not toxic, of course, but once you drink 1 gallon of the water at once, also toxic, they will kill you anyway. So it's depend how you use the nanoparticle. But in my experiencing that the nanoparticle can, of course, can reduce the dose of the drug because more effective, more pronounced when we compare with nanoparticle drug, and then that can, of course, when we want to, like I said before, give it to the patient, we make sure that the dose that we give to the patient is not toxic for the patient. So several tests have to be done before we go to the patient. Thank you.
Okay, thank you, Pak Ronny, for the question. The answer. So I would like to comment to Uzul Hikmah. So is that clear, or you still have questions for Pak Ronny? Okay, it's enough. Thank you, Pak Ronny, for your answer. All right, there's another question in the chat box. It's for Francois. It's from bracket. Please go to Kayo. What would you suggest for the extract concentration we should start on developing antibacterial from plants? Should we start at small concentration, for example, ppm, or we could start on percentages? What factors we should take into consideration? Thank you. All right, Francois, thanks, yours.
Yeah, that's another good question. The question of concentration. Of course, if your concentration that you are testing is too high, you won't have a good result and the effect that you will see maybe won't be the fact that it inhibits the growth or have any other mechanism of action, but just because there is not enough nutrient inside the well. So the concentration is really something important that you have to look at and to define before performing the experiment. basically so there are different guidelines, and we try in the paper that I showed you during the talk, we try to define a kind of breakpoints to evaluate the concentration, and I will say that at least don't go over 1 milligram per milliliter, since even like some, but the interesting point as well is that we have seen that at some stage when you test a 10 milligram per milliliter plant extract at 10 milligram per milliliter, you can still get an activity, and if you do the bioguided fractionation, you get some compounds, some bioactive compounds. But I will suggest to start from 1 to 10 milligram per milliliter, which is quite high. And then dilute the compound by 2 and reach like a very low concentration to make sure that you screen the whole range of concentration that you can get. And so yeah, basically this is it for plant extract. We can define a real good antibacterial activity for plant extract as being below 100 microgram per milliliter for MIC, so for the minimum inhibitory concentration. So if you have a minimum inhibitory concentration below 100 microgram per milliliter, it's we can define a plant extract as being antibacterial, or at least with a growth inhibition activity. And for a natural compound or isolated compound or single compound, we can define an antibacterial natural isolated compound as with an antibacterial activity below 10 microgram per milliliter. So yeah, that's basically what we use as a threshold in antibacterial tests.
All right. Thank you, Francois. Breakfast list to Kayo. So, do you still have any more questions or maybe confirmation to ask to Francois?
That's enough. Thank you.
Okay. Thank you. All right. Thank you, Francois. So we still have a lot of time to have questions to both of these lecturers here. Actually, is there any questions you can ask in the chat box or even raise your hands? Okay. So while we are waiting for another questions, other question maybe, I would like to ask to Francois. You said that most of natural product is antimicrobial, and then the natural product is mostly from bacteria, right? From your in your presentation. So is it from terrestrial bacteria or terrestrial natural product, or is that including from marine as well? Because as you know that the marine is a really good source for antimicrobial.
Yeah, that's a really good question as well. So I think that these the natural product discovered from bacteria and Actinomycetes were found from both, basically from terrestrial organism and marine organism. But we did more research on terrestrial microorganism in the past. So it would make sense that more natural products come from terrestrial microorganism, and since we did not like dig into the marine area, we did not know much about the marine microorganism. There are still a lot of things to discover, I guess. So this is why more research should focus on this particular topic, I would say, or space. And so yeah, that's my answer. Yeah.
Yeah, okay. Yeah, it's really interesting because actually marine sources is not been explored yet, actually, especially there are like bacteria symbiotic, as like a symbiosis with sponges, for example. There's a lot of there's another question in the chat box. I have to translate. I think it's for Francois. It's in bahasa Indonesia, sebagaimana untuk media pertumbuhan untuk bisa menjadi antimikroba yang efektif. It's from Anita Manulang. It's how the media growth media is becoming as an effective antimicrobial. I think I don't know. Is the media becoming antimicrobial, effective antimicrobial, or is it for maybe the media to grow the antibacterial or something like that? So, yeah.
The question was about media, right? The media that we use.
Yeah, the media that you use to be antimicrobial, that more effective, the effective antimicrobial. That's her question. But I think it's maybe the good antimicrobial, maybe antimicrobial media.
Okay, I'm not sure if I understood well the question, but if it's about the media that we are using in the tests, the antibacterial tests, it depends on the bacteria that we are using. So I'm not sure that this is the point. Another point as well is that if you, of course, the nutrients brought to the bacteria will influence the activity and the growth of the bacteria. So this is another interesting point, is that we have seen that when we deplete when we reduce the media and we avoid the use of iron, the bacteria do not grow well, because we know that bacteria need iron to grow. So yeah, there is something interesting beside that, and there is a lot of things to study as well. Yeah, I hope that I answer well the question, and this is what she wanted to hear.
Yeah, I think so. It's from Mbak Anita. So I would like to confirm. Is it answer your question? Jadi untuk media pertumbuhan itu tergantung dari bakteri yang digunakan, juga banyak sekali faktor yang menjadi pertimbangan. Bagaimana Mbak Anita? Ada pertanyaan lebih lanjut Mbak Anita? Is there any more questions? There is no. All right. There's another question. Says it's more for Francois here. First is from Prisi Permanasari.
Should I answer the question, or? Yeah. I don't hear you anymore. Do you hear me, or something went wrong? Or?
Yes. I hear you, Francois.
Okay. So I guess I can answer the question. Yeah, so the advantages of antimicrobials derived from natural products compared to synthetic drugs in terms of therapy. So as for me, we have seen that natural products can provide antimicrobials, but the most important thing is that basically we're gonna use the scaffold formed from the antimicrobial scaffold formed from natural products to make derivatives and to make more powerful compounds and antimicrobial compounds. So it's not that we require larger dose to have a therapeutic effect, but more that most of the time when we use antimicrobial compounds and we test it inside the human body, we can have like some bioavailability issue, and that's why Ronny tried to explain us is that by making nanoparticles we can improve this bioavailability. We can have some toxicity issues as well. So, and so there are like different problems that we face when we isolate natural antimicrobial natural products. And to try to avoid this problem, we need to make derivatives. And so that's also why it takes a long time to develop antimicrobials from natural products, since we have to start from one compound and then make different compounds from that and test these compounds again and make sure that these compounds work well. So yeah, it's a long step. It's a long step, yeah.
Halo Pak Pius. Halo.
Sorry, I haven't unmute my. I just moved from inside to outside. All right. Another question from Hani Fitriani is for Francois and for Pak Ronny. For Francois, how we can be sure that our compound from plant or extract give the specific clinical effect, especially from ethnophytomedicine that used from ancient. And then for Pak Ronny, how do we determine or choose a good nano formulation for our drug or extract? Maybe I would like to give the opportunity first to Francois.
Okay. Yeah, so I'm going to answer that quickly. So basically, when you looked at the traditional medicine and you try to ask to define and to understand how people think the disease, and so there are a lot of ethnobotanical work and ethnoanthropological work to do to define well the different disorders that are present in a community. And based on this knowledge, this very strong classification of disorders in this community, you can do an ethnopharmacological survey and link the plant species, one particular plant species, to a specific disorders. And from that, you're going to select in vitro assay, so specific in vitro assay, and then specific in vivo assay. And of course, that's how it works. Basically, you start from the traditional knowledge and how the plant is used in humans in traditional medicine, and then you go over in vitro and in vivo assay. And you can, if you isolate a natural, a single compound, you can even do clinical trials at the end.
Okay, Hani, so I would like to before Pak Ronny give answer. So how is the answer from Francois? Is it are you satisfied enough, Mbak Hani?
Yeah, sorry, Francis. First from all, so we can follow the clinical effect from them, from the like anthropological location from the extract. Can we follow the clinical effect from the ethnobotanical from the location that the extract or plant claim about the clinical effect before we choose the compound have the specific clinical effect first of all?
Yeah, so I guess that you want to know how we can know more about the clinical effects based on the traditional knowledge. Is it your question?
Yes, yes,
Yes.
yes.
So basically, the clinical effects. So that's what I try to explain, is that first you try to know well the disorders inside a community. So if someone speak about I use a plant for fevers, so you need first to know what kind of fevers, right? To make sure that to define if it's a general fever, if it's malaria, if it is an infectious disorders, and if it's and which one. So you want to know like very precisely how this plant is used and what kind of like I would say disorders can be treated with this plant. And so that's how we work, and we also try to combine different information from different people to make sure that when we get information from one traditional healer on one person, we want to make sure that this application for one plant is also the same for another, and then you get a better idea on how this plant is used. And if someone if, for example, you have 100 people who tell you, okay, I use this plant for treating wounds, okay? So you are almost, I mean, you have like a great, you're almost sure that these plants are effective since it is used by many people, and the same way by many people that do not know each other. So that's how we know that the plants can have an effect, and a very specific effect, a very specific clinical effect. And then you evaluate it by using like a specific pharmacological model, basically. Is it okay for you? Did you understand what I said?
Yeah, I think Mbak Hani is already understand. Okay, now Pak Ronny, please, your time now is to answer the question.
How do we determine or choose the good nanoformulation or drug for drug or extract? I mean, depends of the polarity of your drug. For example, if you have a non-polar drug, they don't like water, for example, they like oil, for example. So if you have like active ingredient that have non-polar, I think emulsion is the good choice. Or you can also make neosome, because with neosome, for example, you put your drug inside the membrane of the nanoparticle. With emulsion also the same, because emulsion basically made from oil, so you can put your drug inside the oil because it's the same, they have the same polarity, for example. But if you have like drug doesn't have drug that have polar or like water, you can make like liposome, for example. You can also make dendrimer. You can also make like polymer nanoparticles. So everything depends on polarity of your drug, because it's easy way to decide it with kind of nanoparticle that we already we want to make. You know, is it my answer?
Okay, Mbak Hani, I would like to ask you, do you still have any questions? Maybe more questions to Pak Ronny?
I think enough, Pak Ines. Thank you so much for the answer.
Okay, thank you, Mbak Hani. All right, we still have what 10 minutes for question and answer session here. So we are waiting for other questions from the audience, from the audiences here. Is there any questions? I think it's a really good, what is the, because Francois is on the sorry for the the background sound. Here we have Francois as an affiliation the antimicrobial from natural product, and then not only support plastic development using nanotechnology. I think if we can collaborate that, so that really good collaboration actually, especially as Francois said that Indonesia is actually more has more resources. We have our country is the first or number one in biodiversity, including the marine. So it still need to be explored actually. So so is there any questions from the audiences, the participants maybe? Maybe I have one question to Pak Ronny. I'm not an expert in nanotechnology. I'm just wondering if we make a have what is a single compound. Yeah, single compound is isolated from natural product, and then we would like to make as a nano dosage form there. So I'm just wondering, is the compound is changed the we have to change the particle size of the compounds, or what we do when we do to make a nanoparticle from from the isolate.
Yes, basically when we working with nanoparticle is we don't we working as a bus. So we create delivery system on it. So the size is, of course, nanoparticle size is larger than active ingredient size. For example, one C atom is like 0.1 nanometer, for example. And then if you have an active ingredient with 10 C, for example, 10 C atom, the size is 1 nanometer. So some people be confused when that nanoparticle is smaller than the active drug. It's not the correct. Nanoparticle always bigger than the active drug. So nanoparticle is basically we make like a vehicle that can deliver or and then also can protect the active site, the active drug to the target that we want. So it's like we create like a bus, like I said before. We can whenever we develop the bus and then the bus can deliver that drug to the target that we want. We can load whatever we want inside the truck, inside the bus, for example. So basically, we don't make changes with the active site, for example, of the drug. We don't change anything. Just put that inside the truck, inside the bus, and then send it. So of course, the size is bigger than the active site, but still in the size of nano. For example, we always create a size of 100 nanometer, for example, because the key is we have to make the particle get smaller than the cell, the smaller than the bacteria, because our target, for example, for bacteria is like 1 micro. So we have to make a nanocapsule, nano particle that smaller the target, so we can easily to target the cell. This is my opinion, my answer.
Okay, thank you, Pak Ronny. Yeah, it's give enlightenment to me, because So actually, it's a nanoparticle kind of a drug delivery system.
Yes.
Yeah. So we don't need to. I mean, like we don't. We change the size of the particle of the active compound, but we have with the what is gold or silver, something like that. Yeah.
Yes.
I think it is interesting, for maybe solve the problem with the difficulties, how the for maybe it's to solve the problem with if there's a problem with the not bioavailability, the solubility usually, yeah,
Yeah.
the solubility of the isolate compound or the extract. So I don't know if in maybe in vitro assay we can using nanoparticle to to overcome the problem with I don't know, non-polar maybe can be as Francois suggests in this diffusion method. There's a problem with the non-polar compound because I think because of the solubility, maybe it can solve that problem.
Yes.
Okay. All right. Is there we still have maybe 1 or 2 minutes actually for the Q&A sessions? This is interesting actually, but I think it seems there's no more questions for the from the participants here, and because of the time limitation as well, I think I would like to finish the lecture sessions this afternoon. But before that, I would like to thank Francois and Pak Ronny for your great and interesting lectures that you gave us today. And it is very useful for us, the information from Francois and Pak Ronny. Before I finish this lecture, we have a token of appreciation that we would like to give to Pak Ronny and Francois. This token of appreciation is e-certificate. Maybe I would like to ask if is there an Bu Suci as a head of the master program of pharmacy. Is there any Bu Suci? All right. So because Bu Suci is not here, so I would like to give first the certificate of appreciation to Francois from PhD as a speaker in our webinar series today. Thank you, Francois, for your great lectures today. And then next, I would like to give a certificate of appreciation to Dr. rer.nat Ronny Martien as speakers in a webinar series in nanotechnology. All right. Before we end the session today, please join me to give applause virtually to Pak Ronny and Francois here for their great lectures today. And thank you once again, Pak Ronny. Merci, Francois, for the lecture, and hopefully that we can meet again one day, either in a webinar series or maybe give a lectures, or maybe Francois, you can visit our campus and then maybe meet Pak Ronny as well in Yogyakarta. maybe because we know that pandemic is already hopefully that we've been becoming a better and better here the situation. All right. I'm finishing my as a moderator webinar series of Farmasi UIE Antimicrobial Development. I would like to say thank you to once again for both speakers, and I'm finished the session with reciting Alhamdulillah. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. I give the time to the MC, Mbak Ilya, please.
Okay.
Time is
Okay.
yours.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much for APT Pinus for your time. Ladies and gentlemen, please give a big applause for all the speakers and also the moderator and all participants who have been attending this event. We have a door prize for the best question, including first is for APT Asih Tri Astuti, second is for Uzulul Hikmah, and then Brekes List, and then Anita M. Manulang, and then Prisci Permana Sari, and then Hani Fitriani, and the last but not least is Anton Hustanto. Please contact the Magister Farmasi UII or Universitas Islam Indonesia by direct message on Instagram at magisterfarmasi_UII. Finally, we have already completed the whole series of event International Webinar Series 1 2022. Representing all the committees, we apologize for all inconvenience things for you during the whole event. We hope this event can be give you a lot of excellent information that can be applied in the future. See you again. I'm Ilya Masyrahila. Wassalamualaikum warahmatullahi wabarakatuh.
Ya, teman-teman semuanya, terima kasih. Thank you, Francois, dan see you again.
Merci, Francois. Thank you. Terima kasih, Pak Roni.
Sama semuanya, guys. Saya izin pamit, guys.
Iya, Pak Roni.
Semua.
Terima kasih semuanya.
you.